Right. All right. Assalamu alaikum wa rahmatullahi wa barakatuh. Good evening, everybody, and welcome to the Saudi Society of Blood and Marrow Transplantation weekly webinar series. Thanks to Dr. Ahmed Al-Askar and the SSBMT team for your kind invitation and organization. I'm Ahad Al-Srehi, a postdoctoral research scientist based here in Riyadh. And it gives me a great pleasure on behalf of the SSBMT to introduce you to our speaker for tonight, which also happened to be my PhD supervisor, Professor Jude Fitzgibbon. And he's going to talk to us today about a very exciting topic, which is the use of multi-omic approaches to better understand and treat acute myeloid leukemia. So just a brief introduction about Jude. He's a cancer research scientist from the Barts Cancer Institute in London, that is part of Queen Mary University of London, where he's a professor of personalized cancer medicine and center lead for genomics and computational biology. His research focuses on the molecular pathogenesis of both leukemias and lymphomas, aiming to improve on current diagnostic, prognostic, and treatment strategies. His research on AML focuses on both sporadic and familial forms of the disease and holds program funding from Cancer Research UK and many others. He's also an associate editor for the British Journal of Hematology and is the chair of the UK National Cancer Research Institute Science Subgroup for AML and lymphoma. Did he also mention that he is a fantastic PhD supervisor and group leader? So a big welcome to you, Jude. But before you begin, I would just like to remind you that tonight's webinar is sponsored by SSBMT and Janssen as well. And also a reminder to our audience to please post your questions in the Q&A box, and we'll be happy to answer them at the end of the talk, inshallah. So without further ado, please go ahead, Jude. Thank you very much. Thank you.
So good morning or good evening, everybody. It's really a fantastic pleasure and an honor to be invited to speak at the Saudi Hematology Series. I'm based in London at Queen Mary University of London and the Barts Cancer Institute. We work primarily on lymphomas and acute myeloid leukemias. And today I want to tell you a little story about the use of multi-omic approaches to better understand and treat acute myeloid leukemia. And I can imagine a lot of you are either treating these patients or doing the diagnostics that run in parallel are important for the multidisciplinary meetings when you're deciding on specific therapies. So just a little summary to start in relationship to hemophoresis and what acute myeloid leukemia is. And just to really say that it's a stem cell disorder. And so this is your hemophoretic stem cell. And typically you get a block in differentiation, you get increased proliferation. And that leads to the accumulation of immature cells called blasts, which clog up the blood and essentially gives it its acute nature. That's as simple as that, really. And so in a way, a lot of the work has gone on in identifying what are the key genetic mutations or the key genetic events that are driving either this block in differentiation and change in proliferation. And as Ahad has said, my group work on both familial and sporadic disease. And, we've got several different publications in the area. And, really Ahad's work focused specifically on this family. It's a GATA2 pedigree. And what you can see here is that you've got an asymptomatic third generation where none of the individuals were affected, but they were carriers for the GATA2 mutation. While in the fourth generation, a lot of these individuals were actually affected. And Ahad was able to then define specifically what was actually causing that distinction between the asymptomatic and the symptomatic individuals. And that was silencing of the GATA2 lockers. So that's one area, and I think Ahad has really made a major contribution to that for us. But what I'd really want to talk today about is probably a greater problem that reflects maybe 95% of patients with acute myeloid leukemia. And you'll all be familiar with the journey that a patient has from diagnosis to, unfortunately, either cure or disease relapse, with allogeneic stem cell transplantation clearly being an important therapeutic approach and getting patients into a remission, and then getting patients into a durable long-term remission, and hopefully cure is key. And all these areas offer an opportunity for new therapies and new ways of treating the disease. And we definitely need new therapies, because this is a survival curve from the MRC trials. And you can see that from a cytogenetic point of view, where probably we've had this kind of information for the last 30 years or so, we can subdivide patients based on their initial diagnostic translocations, whether that's the 15, 17 here. And then everybody is probably aware that, from a cytogenetic point of view, if you present with complex cytogenetics, with monosomy 7, monosomy 5, some of the MLL translocations, your outcome is particularly poor. And I think of late, what's happened is we've started to subdivide patients subsequently based on additional gene mutations. And these mutations, generate, I think, more complexity. Clearly some mutations like CYP-alpha or NPM, IDH, P53 are informative, but I think the cytogenetics does quite a good job. And I think anybody who's treating, the clinicians, the hematologists that are here on the call today, and people working in diagnostics will all know that outcome is very poor, and that things really haven't changed materially in the last 30 or 40 years. And one way to think of that is that we all like the notion of precision medicine. We all like the notion that, well, you've got a personality trait. The cancer has a mutation or a translocation or a change in its biology. And if you actually target and revert that change, then you should get a cure. And we all know that's not the case, because targeted therapy, so for example, FLT3 inhibitors, great idea, but actually they don't work so well in that everybody with a FLT3 ITD isn't going to go on and go into remission and be cured. Indeed, actually, quite few longer term are going to be cured. And that's because I think we have to think of AML, and we have to think of all cancers as having primary lesions, but also a lot of modulating changes. And these changes can actually make these effects stronger or provide ways by which the cell can become resistant to any therapeutic intervention that we might try. So I'm Irish, so we talk in very general terms, but you can think of AML from a personality point of view. Think of the disease as a person that we want to try and better understand. And so in a way, measuring a single network isn't really going to be sufficient to understand the disease and be able to fight against it. If anything, we need to have a better and more accurate prediction of the pathways that are important, which ones are active and which ones are important in therapy and resistance, with a view maybe that will actually change how we approach the disease going forward. So it's a very simple and probably uncontroversial hypothesis. Multiple mechanisms are responsible to promote and sustain poor risk AML. Nobody's going to disagree with that. And focusing on a single deregulated pathway or actionable mutation will not lead to improvements in overall survival. And that's quite true, because we all know from our own personal experience, we're individuals. If you just target a single personality trait that we don't like, it's not probably going to make a lot of a difference to us. But if you start tackling multiple different personality traits or key things that are important to us as individuals, that will have an effect on us. And in the same way, maybe we believe that's what's going to happen in acute myeloid leukemia. Now, because the world is a very competitive beast, we sat down about 2 or 3 years ago and we said, what area of AML do we think is most in need of research and support? And what we decided to do was to focus in on poor risk AML. And this is data from the MRC clinical trials, 13,000 patients, of which over 2,000 are poor risk. And so I'm sure that the data is very similar across the world and is very similar in Saudi, but about 40% of patients outside of the poor risk group will be alive at 5 years. And there you see an age effect where for patients with poor cytogenetics, as I said before, the monosomy 7s, the complexes, the MLLs, the outcome can be quite poor at 14%. But equally, 6% of patients over the age of 65 will be alive at 5 years. So it's really the worst of the worst in relationship to hematological malignancies. So what we decided to do then was to build up a patient cohort of patients that have been treated at the NHS Centre Barts Health NHS Trust. Patients would come in, you would take their bone marrow or peripheral blood sample for diagnosis, of course, and cytogenetics, but any extra would be stored down. And then you could follow the clinical information on the patients, the cytogenetics, and have all that information available and viable cells. But rather than thinking of just looking at genetics, we said, let's see, can we build a really deep personality for this AML? Let's do RNA-seq analysis, sequence the transcriptome, do whole genome sequencing, targeted resequencing, and I'm sure targeted resequencing is part and parcel of the diagnostics that you're doing in your hospitals in Saudi. But we're also adding proteomic analysis, phosphoproteomic analysis looking at cell signaling, CyTOF analysis, that's a fancy flow whereby you can look at 39 or more markers. And critically, in conjunction with a group in Finland, we're also doing drug screening of these cases. So in a way, we're building up a big picture of each individual AML. We have the clinical information, but now also we're building up a much greater notion of the personality of the cancer itself. So what we're really saying is dissect the genetics and biochemical features of poor risk AML. And maybe we'll be able to stratify, identify maybe some patients in certain groups that could be treated differently and design better therapies, just using this multi-omic data. Now, I'm not going to go in this evening into the individual details, but clearly you do need samples whereby we've collected a lot of material on a patient at diagnosis that the patient would have a high white count. And then you need to store the cells in such a way that they're viable, that they can actually grow in culture afterwards. And we've been very fortunate at Barts over the last 20 years to be collecting fresh material from patients, storing them in liquid nitrogen so that we can then ask our biological question, find the vial that corresponds or the vials that correspond to that individual patient, and then manipulate the cells either for DNA and RNA extraction, for proteomic analysis, for flow cytometry, or for drug screening where those cells are grown in culture. And so in addition then to the proteomic analysis, we're doing CyTOF analysis, which is just a way of getting measurements of different cellular populations that are present in the AML itself. And these are some of the markers, whether you're focusing on the blast, lineage-associated markers, or other aberrant markers. And altogether, as you'll see later, this kind of information is important in deciding whether there's the AML, the leukemia is a primitive one, or maybe it's a more progenitor, a more mature-based AML. I think people would be probably more interested in the ex vivo drug testing. And so in a way, we can provide samples to our Finnish collaborators in Helsinki. And they can then take those cells, dish them out into different wells, and we can test for the majority of the cases we have, about 50 poor risk AML cases. We can test about 500 or so different drugs. And those drugs can fall into different groupings. Some are already licensed for therapy in other cancers. Some are novel therapies, and others are very experimental. And this pie chart here just kind of really breaks down the nature of the different types of drugs that we're testing, and so that they come in many different flavors, many different types. But altogether, you can see how we can build a very large volume of data for us. So where are we now, 2 years, 2.5 years into the project? Well, where we are is we've now got our data, and we've got all our omic data on those 58 samples. And I want to tell you 3 stories to give you a kind of a flavor of how we're utilizing that data and seeing whether we can improve treatment or propose new treatments or to develop some novel clinical trials that would focus in on certain groups. or because these drugs can be toxic, whether we can identify which patients are most likely to benefit from certain therapies. So I'm going to focus in initially on a story about the 6/9. It's a rare translocation called DEC-NUP214. I'm going to talk about cell composition and drug sensitivity, which I think is really interesting. It's a new idea that's coming through in different collaborating groups at the present time. And also I want to show you how we can kind of align specific inhibitors and specific signatures in the AML as to which patients may or may not respond well to certain therapies. And we've got a lot of choices, but I'm only today going to tell you about MDM2, but there's lots of others that we're actually working on at the present time. So the 6/9, in the 58 patients, this is rare. So a lot of the cytogeneticists or pathologists, morphologists, clinicians are going, oh, these are very rare. Well, and they are rare. Probably about 1%, maybe 2% of all acute myeloid leukemias. But they are interesting because a lot of the patients are younger. So you can get pediatric cases, and then you can get younger adults. And as you get older, like I do, your definition of what age is actually changes quite a lot. So I'm around the 50 mark. For a lot of patients around 50, 40, and even younger. The 4 we have here, what's also notable is outcome is very poor. And patients tend, in addition to having the DEC-NUP translocation, also have mutations in FLT3. So they also have a FLT3 ITD. And it causes a translocation between DEC and NUP. And very little is known about this translocation, aside that the patients do poorly, and it coincides with FLT3 ITD. Now, this might scare some of you, but I'm just going to give you a little flavor of the type of information that we've created. So this is RNA-seq. This is looking at the transcriptome of AML. And these are the 4 6/9 patients. And this is an unsupervised analysis whereby we're looking at the gene signature for all the different AMLs, and then we're asking ourselves, is there something special about our 4, the 4 with the DEC-NUP? And there is. They cluster together and they have a very defined signature. And then what we've done is we've worked with a group in Canada, Guy Sauvageau's group. And what they did is they put some of their mRNA data with our mRNA data from the cases, and then compared them to several hundred other AMLs. And so what you can do then is you can identify key genes that appear to be highly upregulated in the DEC-NUP cases. So it's possible that the translocation in some way is actually driving an increase in these genes. And these genes then become potential downstream mediators for how DEC-NUP might function. Now, what's important to keep in mind is, although these individuals have FLT3 ITD, there's no link with FLT3 ITD in these genes. So it appears that these 5 genes are key potential mediators in relationship to the 6/9 translocation. And we're now looking in hematopoietic stem cells to silence these genes and see whether it has an effect. But on top of that, we've got the in vitro drug screening data. And of the 500 drugs, what we saw is, we saw the drugs that showed greatest sensitivity in these groups of patients were the XPO1 inhibitors. Now, a lot of you are probably thinking, one, they're very toxic drugs because they're licensed for multiple myeloma at the present time, and patients, they're hard to tolerate. But XPO1 came up very highly, as did ruxolitinib came up highly in the screen, as well as in vitro sensitivity. And so giving us some clues as to whether we ask the question going forward, is there a link between these drugs and any of these genes that appear to be upregulated? Also, I think when we went to look at the XPO1 clinical trials, what we could see is that there was one patient with 6/9 included in the XPO1 clinical trials, and that patient went into a durable remission, suggesting again that maybe this is a therapy that could be personalized towards this group of individuals. And then, of course, because we need to generate in vivo data, we can take our primary cells and we can engraft them into patient-derived xenografts. That's good, because then in a way we've got mouse models of 6-9 that we can then test these hypotheses and see whether the depth of translocation, we understand a bit more about the biology, but also we can understand a little bit more about the therapy. And then to pull together these groups of patients, probably internationally because they're rare, to see whether our therapeutic approach is valid or not. That's the first story. Now, the second story is probably something was very new to me about 6 to 9 months ago, and it made me quite excited. So everybody around the world, hears about single-cell RNA-seq. And so this is a way of looking at the transcriptome of the individual constituents of the bone marrow, peripheral blood, AML, any different cancer you want to look at. And rather than getting a kind of an average of the bulk, you're now looking at individual cells. And so what you can do then is you can take those signatures and you can say, okay, well, this is what the stem cell signature looks like. This is what a GMP signature, a monocyte, a dendritic cell signature looks like. And then you can start creating those signatures and using bulk RNA-seq data from your primary AML samples and saying, patient here, well, it looks like patient A has signatures across all the different cell types. Patient B, that looks a little bit more like a hemophoresis stem cell type, a more primitive AML, the blast population, because we're essentially taking bulk, which will be the blast population. You can start subdividing patients based on their maturity. And this is kind of another way of looking at it. So you can generate single cell signatures of how each cell type is talking, and then ask in the AML itself, how many of different cell types, or what's the kind of composition? Is there a bias towards either primitive or more mature cells? And then you can build up a profile of the patient. And this is work that John Dick presented at ASH and something that we've done, but I don't want to take credit for the idea because I wanted to give the credit to him, but I'll show you our data as well. So you can see that you can take different series of patients. This is the TCGA cohort. You can see that this case, it's not completely primitive, but the predominant signature in the mRNA expression is of a primitive stem cell origin. This is GMP, this is intermediate, and this is mature. So not only can we divide patients now up based on their cytogenetics or based on their mutations, but you can also break up patients into different groups based on the maturity of their blast population. Why is this important? Well, it's important because different AMLs may respond differently depending on their level of maturity. So the more immature cells, the more primitive the population, there's evidence to say they're more sensitive to the venetoclax, to the demethylating agents, etc. While the more mature might be more responsive to other therapies. So you can see you're layering additional data onto who or which type of patient might respond best to certain therapies. Very important. And really, it shows that you can squeeze out more data, and also that it's more complex than just have you a mutation. Mutation is the start, and it's an easy story to put down on paper and say, poor if you have FLATE, good if you have a biallelic CYP alpha, etc. But now we're adding extra layers of complexity that I think will be important going forward as we try and personalize and give each patient the best chance of responding well to the therapies we have and to develop new therapies. Now, it's important, of course, to think not only in cell composition, but also in relationship to the proliferation of the cells, the survival, and only 6 of the cases that we've tested in our 58 are alive at the present time. And of course, age of diagnosis will be key as well, because we haven't discriminated between under the age of 65 or over. We've taken cases where we have biopsy samples available to study. And, you know, in a way, somebody is probably saying, but this sounds like the Fab type, doesn't it? You know, we can break up patients based on the Fab type. And in a way, RNA deconvolution provides a kind of a classification, but actually with a little bit more nuance, a little bit more detail. That's probably more important. And so this is our data where we do exactly the same. We only see primitive, mature, and intermediate cases that we can pull together. But the primitive cases are very clear. And you can see here, there's a mix of karyotype irrespective of that enrichment in relationship to one specific primitive or a change in maturity. That is not divided based on cytogenetics alone. And what we can do then is we can take the data that we have on our cases, and we can ask the question across the 500 drugs we've tested, is there a differential response based on the data that we have? And probably we're one of the few groups in the world that would have both the RNA-seq data and now the drug sensitivity data to kind of ask these questions. But we also on top have the CyTOF data, because the CyTOF data, the flow cytometry data, probably allows us to make subtle changes in our definition of primitiveness versus maturity. So we can follow some of these new drugs, and the plan really is, and the model that we're taking is then to talk to different biotech companies who are either exploring certain drugs or certain therapies, or are running phase 2 or phase 3 clinical trials to see whether, you know, we can help them in deciding which patients are more likely to benefit from these drugs. Because, you know, as we all know, a lot of these trials don't get past the early phases because they fail. But it's possible that there might be a population of patients that could do better on those therapies if we could pre-select those for study. So cell composition and drug sensitivity. So a little summary. So RNA-seq deconvolution. So you can use the expression, the gene expression, the RNA transcriptomics that are present in a cell to deconvolute and get an indication of dividing AMLs into different groups, not along the lines of cytogenetics or mutation. Mature patients appear to do better than primitive patients. It's a little piece of additional information. And clearly the drug screening suggests that there is a bias one way or the other, depending on whether you're primitive or mature. And I think it's important to say that you know, we've jumped on the bandwagon here a little bit, and that data is very much linked to John Dick in Toronto and other groups. But it's a new idea that's coming into the literature, and now we've got an opportunity because we've got the primary data to ask these questions. My last story. So in a way, MDM2 inhibitors have been around a long time because MDM2 is a way of turning off P53. P53 is kind of the guardian of the genome, as people say. And anybody here would know that, or I would know myself, if I had acute myeloid leukemia in the morning and P53 mutation was present, I would probably say this is very, very bad news. I probably have a complex karyotype, plus I've got a P53 mutation, and so I fall into the very, very poor outcome population of patients. So MDM2 inhibitors are a way of reactivating P53 in a way then trying to just push the cell towards apoptosis. And so in the panel of drugs that we looked at, there's 4 different drugs, 4 different MDM2 inhibitors. Idanutlin is a Roche drug that's failed phase 3 clinical trials because of toxicity. That's my understanding. And the other drugs are in various stages of clinical testing at the present time. And in our samples, what you can see here is that the blue signifies a very high correlation of responders, non-responders across the 4 different drugs. So this is good, because it shows that our in vitro study is actually working well. And we're getting very robust data because we're getting very similar results for the different available MDM2 inhibitors that are present. This is just a kind of a little summary for us here about how MDM2 works. But essentially, by getting rid of MDM2, it can allow activation of P53, and having activation of P53 is a good thing. Now, this isn't hopefully it's a very simple slide, and so I'm going to walk you through it very slowly. So as you said, we've got a very homogeneous response to the 4 MDM2 inhibitors. The key thing is, if you have a p53 mutation, you do not respond to the MDM2 inhibitors. So these are our responding samples. We just picked the extremes here to show, and these are our non-responding. And the gray bars are the p53 mutated cases. So you can see if you have a p53 mutation, then in a way, in our hands, nothing isn't going to work, or MDM2 inhibition isn't going to work. And that's important because that allows you to say, if I'm running a clinical trial, I shouldn't incorporate patients with a p53 mutation. And up to now, most of the studies have incorporated all comers. And not surprising, the outcome across the board probably hasn't been very good, but you should certainly not, I would say, include p53 mutated cases. And you can see here, this just shows the 2 different drugs and the differences in response in vitro. Always key to remember this is in vitro, it's not in patients. And p53 status then significantly determines the response to MDM2 inhibitions and represents, in our hands, a first pass of a biomarker. So in a way, what you're saying is the patient comes in, you get back to cytogenetics, you get back to mutations, and you're thinking, where might this patient go next? Then you can probably say, well, this patient has a p53 mutation. The VAF levels, the dose of the mutation is very high. Let's not consider it for anything that's p53 related. So what we did instead was we started to look at the wild type p53 cases, the good responders versus the poor responders. And then you can look into gene expression data, you can look into mutational data, you can look into proteomic data, and you can ask the question, is there anything special that's actually driving failure of these MDM2 therapies in vitro? And what we were able to see is that all the responding cases had a very low p53 signature, gene expression signature. It was almost like as if the brakes were on the p53 pathway. And so the MDM2 inhibitor then in some way released that brake potentially, because we got a very strong association between this signature of 16 genes across all 4 MDM2 inhibitors. Now you'd say, well, that's good, but it's small numbers. I'm sure you're probably saying this is very small numbers. But the beauty of working in collaboration, working together as a group with our Finnish collaborators, was that they had over 150 patients with AML that they had treated on the same drug panel and had some gene expression data. And so what we were able to do then is ask the question, is our signature informative, not only in poor risk cases, but in other cytogenetic risk cases as well? And it was. So in a way, we got a very strong correlation between a p53 index and also a second index that we identified using cell cycle proteins. So in a way, we feel that this is a good starting point to say that if you're using MDM2 inhibitors, a patient at diagnosis with this particular signature is probably more likely to respond well. Now, clearly, there's a toxicity issue in some of these drugs too. But we're talking of an exemplar for all the other drugs that are actually within the panel that we have at the present time. And so what we're starting to do is to actually look now and see, by treating our cells on an individual basis, adding the inhibitors, and then completing RNA-seq post-treatment, we're starting to see, can we identify what are the downstream mediators that could be mediating the MDM2 effect, and whether those mediators might potentially be better therapies going forward. So there are the 3 stories. So you can see, by having the samples available to us, doing a very deep phenotypic analysis, we can now start pulling the individual data together to see whether maybe can we treat 6/9s better in the future? Is primitive and maturity an important factor in addition to mutations that people need to account for or might be informative? Whether in vitro drug screening might in some way help identify in vitro sensitivity and patient populations that are likely to benefit from experimental or existing drugs that mightn't necessarily been licensed or mightn't have shown sufficient efficacy across all patients to actually go forward for full phase 3 or for licensing purposes. But I think the strength is going to be, and Ahad has told me that she's starting to do some informatic analysis herself. And this is something that we're now working with a group in Birmingham on, is artificial intelligence and machine learning. Even though the numbers of the cases we have are quite few, where we have big data, we can mine the data, and then we can ask the question, oh, can we identify new knowledge? Because you could probably talk to every hematologist here, and we could look at the gene expression data, the mutation data together. We could sit around and have tea and think about which drugs look good, which mutations look good. But that's based on our brain power. What we really need is a computational power to take the data and see, can we identify something very new and very different? Pulling together the 9,000 phosphokinase sites that we've identified, the 4,000 or 5,000 proteins that can be well measured using mass spec, the CyTOF analysis, gene expression and mutation and cytogenetic changes that we see. And so this is ongoing at the present time. And it's likely to fail, but it's the right question. And as I said, across all research, I think it's about asking the right questions and then seeing, can you make sense and can you propose new therapies that can be tested in vitro? And then if the preclinical data looks good, hopefully there'll be effort in taking it forward clinically. And this is the kind of features that are now being looked at from a DNA, RNA, proteomic, drug and CyTOF analysis to see, can we pull out some new data and new information. Just me finished. So hopefully you've got a little story about the kind of work we're doing. We haven't arrived yet. We're halfway through our funding from Cancer Research UK. And we've got a team. So Finn Copley and Anna work with me. We lead on the project. But we've also got a lady called Dominique Bonnet. Many people will have, she's a stem cell biologist, did her PhD with John Dick. She works in the Sir Francis Crick. David Tausig is head of hematology at the Royal Marsden. Jamie Kavanagh is head of pathology at acute myeloid leukemia at Barts. And Jean-Baptiste Cazier is head of computation in Birmingham. We also work with a Hungarian group, Csaba Butters group. And I think the drug screening data that we have from Kimmo Porka and Caroline Heckman in Helsinki is key. It adds so much additional information and so much more correlative science, and that is of interest to biotech, but also interested from our perspective to see, can we translate any of our observations going forward. So that's it. And as I said, hopefully in the next couple of years, you're going to see some really nice publications suggesting new ways of which we can actually treat this group of patients. Thank you.
Wow, that was wonderful talk as usual. Thank you very much, Jude. We really enjoyed it. I have to say the acknowledgement slide made me feel a bit nostalgic because, you know, I know those people, some of them, and I have worked with them. So yeah, a reminder to our audience to please type your questions in the Q&A section. We have some questions here, but to hit the ground running. So yeah, your talk, Jude, really highlights the importance, I think, of collecting patient samples, right? And dividing patients into different subgroups based on their genetics, and that would help us then select the best treatment. So after generating all these data and integrating the various multi-omics, which I think is the trickiest part, where do you think, or how do you envision this data will take you, say, in 5 years? Like, would it, for example, lead to a clinical trial generation or for these group of patients? Where do you think?
So in a way, you know, you can see the therapy for poor risk AML at the present time is probably restricted to the use of venetoclax and demethylating agents with some opportunity for bone marrow transplantation. I can see Dr. Alaskar is here, and I think, you know, he could comment on the treatment for poorest cytogenetic groups of patients. is really very difficult. And some of these patients then would be elderly with comorbidities. I think what we're hoping is that we will be able to treat individual groups a little bit better. So maybe there's going to be something quite novel to come from the DEC-NUP approach. Maybe there's some new biology there that will be of specific interest. And it's an exemplar. We need to show that this approach works, that we can identify new therapy, and then that can be tested. You can probably select patients that are most likely to benefit. We're also talking ahead to a number of biotech companies at the present time who have some of those licensed drugs that are on the panel. And we can generate these signatures like we've done for MDM2. And we can go to them, and they're very interested because they're running new clinical trials. They're collecting the material as they're running the clinical trial. And what we're trying to propose to them is, if we have the RNA-seq data, or if we have the proteomic data on these samples, then we might be able to say which patients are most likely to have responded when you've completed the study, so that they can see whether these tools become good biomarkers for selecting patients for individual treatments. And I think in a way that the last thing is hope. I think that it must be very, and I'm a scientist, but I've worked with clinicians for a long period of time, so I know the areas quite well. But it must be very heartbreaking to be sitting in front of a young patient or an older patient where you know that the genetics is suggesting this patient is likely to have a lot of toxicity, but no therapeutic benefit from the treatment that they've been given. And so hope is important. And I think that there's a lot of new ideas coming through now that hopefully would introduce new therapies. And I'm sure Dr. Alisker would say that, you know, the last 2 or 3 years has been a little bit more encouraging with the IDH inhibitors, with venetoclax. But generally speaking, these are buying time. And I think FLT3 inhibitors so far have been a disappointment. Maybe the new therapies will be better. So all in all, I would say I'm encouraged. I just feel that the kind of data that we're generating in poor risk will hopefully identify some new clues that will be tested experimentally, tested clinically, and lead to better outcome for at least some groups.
Agreed, absolutely. And because, you know, these tumors are complex, right? So there are multiple, you know, pathways intertwined together. So hopefully in the future, rather than targeting a single pathway, we can target, you know, multiple pathways together. So. And
I think that the combination approach is going to be very important. And you know, I think in lymphomas, you know, there's, for the lymphoma doctors here, you know, there's a push to try and get away from CHOP towards lenalidomide and non-chemo regimens. I don't think AML is ever going to be there for a long time. But I think that in a way, if you could reduce the toxicity and you could get the right combinations of different drugs, I think it would be marvelous. And I think that even good quality of life for a period of time, you know, in your 60s and 70s, offers quite a lot. And so that there's a big opportunity here for research to actually deliver something empowering to AML, and then to serve as an exemplar for how maybe we could think about other cancers too.
Absolutely, agreed. So we have a question or comment that very interesting finding. Sounds we're back to Fab classification consideration. Was there any association between cellular composition and response to immunotherapy in AML, such as checkpoint inhibitors or BiTEs or other monoclonal antibodies? So
the first thing to say is that some of those inhibitors mightn't have been necessarily on our in vitro panel to start. I don't think they've come through as being in the top 10 or 20 of the drugs that we've got the greatest response. But as you said, when you delve down into that data, it's possible that a few cases would have responded particularly well. And I think that I've found over my career in research that identifying these rare cases, these uber responders, or some either a family or an individual that appears to have responded fantastically well to a certain therapy, there's a story there that we need to follow. And so I agree entirely with you that all these new therapies are interesting. I think the clinical data is still being weighted in AML, and I don't think we're there quite yet. I think venetoclax is way ahead of everything else from a therapeutic perspective. But what a lot of companies don't do, and I'm not sure what the story is in Saudi, but you run the clinical trial, but you don't invest enough time in these rare responders, or the example where the physician might have seen 5 or 6 patients not do very well on a therapy, but one individual might do so. And to actually be singling out those responding cases, either with stable disease or in some way a kind of a mini remission of sorts, and to try and work out what's going on with this patient. But going forward, hopefully, some of these new therapies, whether they're the bispecific antibodies, whether CAR T cells, there's so many different new therapies on the block. But I think AML is very tricky, really tricky.
And I found that the 6/9 translocation data was really interesting, because I know that some of them are younger and they're really rare. Would FLT3 inhibitors, for example, work in those type of patients, because some of them have FLT3 mutations, or not?
No, so you would think that FLT3 inhibition would be something that would be included as part of the treatment for these individuals. I think about 80% of patients with 6/9 have these have FLT3 ITD, and probably at a high dose, over the 50% or more. But that doesn't seem to be sufficient. And I think that there's new FLT3 inhibitors now coming through, but the survival advantage of FLT3 alone, is probably less than 10% from the data that I'm familiar with, which is primarily the early New England Journal of Medicine paper. And maybe new FLT3 inhibitors will actually show greater efficacy. But it's not the great success that people hope for, because 30% of all AMLs have a FLT3 ITD. And so the question really is combinations of FLT3 ITD inhibitors with something else. And clearly we're missing a trick. And that's why I think genetics alone, I think a lot of people would say, well, have you got a genetics? Can we do a whole genome sequencing? And in the UK at the present time, we're now hoping that every patient will have a whole genome sequencing done on their leukemia. But it just gives you a snapshot of the disease. And I think that we need more information, at least from a research perspective, with a view that we'll get some new clues as to how we might be able to treat either individuals, or even better still, the whole gamut of AMLs in one go with a certain combination of therapies.
Yeah, absolutely agreed. I think, your talk really highlights the importance of employing these technologies to better understand the biology and complexity of the disease. So we have a question here. Do you think the current turnaround time of the current multi-omics would give results in a timely fashion to be of clinical use?
Really good question.
Yeah.
And so in a way, I'm sure every physician would say that if the information is important enough so that a clinical decision can be made that will actually improve outcome from an individual, a way will be found in doing it. So the turnaround time, there's new data out about whole genome sequencing data being available within 36 hours. And so that information will be there. I think clearly all patients will be going through an induction phase and a consolidation phase. And it's really a case of maybe what are you going to be adding a little bit later to treatment. But my view on all these things is that the pipeline can be created if people can see that there's clinical value in treating patients more effectively. But you're quite right, we're not there yet. And you need to see the proof up front before you will do the investment that will actually make that difference. But I think in a way we have to go away from doing the things that we can do. So you can do morphology, you can do cytogenetics, you can now do gene expression and probably mutation analysis. But if that doesn't do enough for you in relationship to developing better therapies or identifying patients that are more likely to benefit from a certain type of therapy, then we need to think bigger, rather than just doing the same old thing. And but it's a challenge, and it means financial investment. I think the other thing to say is the collecting of material is very expensive. We collected material over 20 years. 80% of the samples that we've collected across all our diseases were never used because the standard of care would change. So, we have samples pre-rituximab in diffuse large B and FL. They're no use anymore because the standard of care has changed. In AML, standard of care hasn't really changed so much. And because it hasn't changed, the samples we've collected for 20 years can be followed through into the kinds of studies that I've just described.
Wow.
Yeah. If I can make a comment here.
Yeah, sure. Go ahead, Dr. Ahmed. Go ahead.
Well, a great talk, Jude. This is very interesting, and definitely shed the light on this complex disease. And as you're aware, we haven't made major progress in AML treatment as compared to the other malignancies, I would say, hematological malignancies. And I see, the complexity in that, and the disease makeup and the different subtypes of the AML. Probably we have multiple subtypes. We're not dealing with one disease. However, and looking at, trying to, sort this puzzle, I would say, how about approaching parallel, I would say, having this approach of, testing medications ex vivo as a drug screening for each patient. How do you see how feasible this approach, and how do you see it being, practiced, and what do you see the challenges in this approach? And then, once you get something that's working, then you go like backwards, trying to, identify the mechanism and
no, I think that what you've just proposed is really what I think Kimmo Porkka and Caroline Heckman were hoping to do in Finland, whereby, because it's a small country, because there's and this is what you have in Saudi as well, because in the UK and in America, you've got so many different hospitals treating AML. Well, I can imagine there's a centralization, so that in a way you can orchestrate these kind of things to be done more effectively, whereby you can make sure that samples are collected, that the right volume of samples are collected, that the time between when the sample is being taken to when the drug might be tested or the panel might be tested can be more, in tune with keeping the cells as fresh as possible. But I think what Caroline and the Finnish group found hardest is getting buy-in from biotech, because clearly you're testing lots of different drugs. Some of them are licensed, some of them aren't licensed. There's ethical issues as well. There is turnaround time, and then there's an interpretation, and there isn't a kind of a commercial value to that approach, at least in the beginning. But I think there's 4 or 5 other groups around the world doing something similar. But going forward, I think it's if we don't find in the next short period of time drugs that are going to work well, I think what you propose makes perfect sense, or at least to test it out and see whether you can actually make a difference. Get the risk reward right, and I think maybe there is an upfront investment required, and then hopefully something new would be spotted. So I think what you said is very true. But the turnaround time, I think, is practical if people feel that there's there's value to the patient in providing and prescribing a drug that's most likely to benefit them. And I can imagine as well that, you know, the cost of transplantation is quite significant, not only to the patient themselves, but also financially. And so that risk reward can potentially be balanced in relationship to the research cost over and above the cost for taking care of a patient.
And despite even that, I mean, transplantation doesn't always work in, you know, in AML. You will get those, at least 50% of the cases who would fail the transplant.
Yes. And as somebody pointed out to me recently, and it had never struck me, but in a lot of cases, some of the patients you're transplanting are cured. You just don't know that they are cured, and you feel that the risk is worth taking them forward to transplant, even if they're potentially cured. And so there's a lot of new work to be done to make sure that patients are managed as effectively as possible. But you're quite right. Transplant works, but it's a lot of patients are going to relapse subsequently, and it's not good for older patients.
Yeah, and the tricky bit is that these poor risk AML patients, they continue to evade standard therapy, right? Like, so they continue to not respond to these standard therapies. So we need to come up with a good treatment approach, hopefully.
Sure.
Yeah.
And I think it's, you know, 2/3 of patients are over the age of 65, and so that sometimes when you read papers, you would almost think that AML is a disease of the under 65s. But 2/3 of patients are over 65, and clearly they're going to be less likely to be able to tolerate aggressive therapies, and they need something that will control the disease while actually keeping toxicity at a low level.
Okay. So I'm wondering from the diagnostic perspective, how do you think the diagnosis in AML has changed, you know, with the current genetic tools or the ones that are coming up, you know, that have come up, you know, in the last few years?
Well, I'm sure at the multidisciplinary meetings that in Saudi, it would be the same as everywhere else, that now, in addition to the clinical and morphology and cytogenetics, you would have an NGS panel. You'd know the FLT3 status, the NPM status, the CYP alpha status. You'd probably know the dosage of the FLT3 ITD. And that provides you then with a starting point in deciding what's the most appropriate approach and treatment for that individual. I think going forward, things are unlikely to change. You know, a lot really depends on the clinical trials, the standard of care in an individual country, and then the kind of experimental therapies that are being proposed, whereby in the MRC, you've got the different MRC clinical trials, and then patients stratified into different arms to see whether one type of therapy is better than another. I think the other thing that's changed as well, and maybe this is something for everybody to think about, COVID has changed a lot, because COVID meant that you couldn't just think about research, you had to act quickly. And I think what the world has seen, and in the UK has seen, and I'm sure it's the same around the world, is there's a problem, you need to solve it, and you need to throw money at the problem. You need to act quickly. You don't have a year or 12 months or 18 months before you know whether you've got funding or not. They funded projects very quickly because this was a world pandemic. And I think going forward, there's a lot of lessons to be learned there, where I think governments, and the UK government, I would say, is one of those who will be trying to change the dynamics of research, where they will want to see, can they decide what the question is, they decide what they want to fund, and they will make funding decisions very quickly to see whether they can replicate the success of the vaccines in relationship to COVID and the type of research that was done. I don't think that's going to be easy in cancer, but I think it's a different change where hopefully it will make researchers think a little more deeply as to how can they translate their observations, rather necessarily than just publish them. The translation becomes the key driver, and I think that's the case for patients and for families, and probably now for government funding.
Yeah, absolutely agree. I mean, I hope really we can, adopt these technologies that you have mentioned and you have showed in your slides for our patients here in Saudi. And we do, I believe, like a huge component of the Saudi Vision 2030 and the national digital transformation that's currently ongoing is improving healthcare for our patients. And indeed, one of the applications of the Saudi Genome Program is screening patients for these actionable hotspot mutations and then offering them, therapies based on their underlying genetic profile.
Absolutely.
And also, harnessing, the power of AI, machine learning algorithms. These are all important, to predict, for example, treatment response or the risk of developing these diseases in the first place. So yeah, I really hope that we see transition towards precision medicine over the next 5 to 10 years in our hospitals and research centers. Of course, nothing comes without certain challenges, but I think, having a team, a multidisciplinary team of clinicians and scientists, we can hopefully,
The beauty you have had is that you've got 30 years to see this happen. I've got another 10 or 12 years in research. So in a way, the clock is ticking even stronger or faster for me. I'm sure Dr. Asghar is thinking the same, that in a way, you've got a length of time in your life that you can try and make that difference. And so what I say to everybody here of whatever career stage you are, that if we're not able to do it ourselves, the important thing is that we train people who are actually going to do it for us, so that we can, in some way, our legacy will be that we have identified and supported good people who actually can make that step or that jump that we haven't been able to make ourselves. I feel quite strongly that the world is getting closer.
Yeah, true.
Absolutely. That's great. I hope that, we can do that in the future, inshallah. So it's been a great discussion. Thank you. Thank you very much, Jude, for this excellent talk and discussion. And thank you, Dr. Ahmed Al-Askar, for this kind invitation and organization. And thanks to our audience for your participation and for your attendance. I hope next time, Jude, we get to invite you in person to come in Saudi in the winter, not in the summer. So yeah, and thanks to our sponsors. So yeah, just a reminder to our audience that we have the webinar, the SSBMT webinar coming up on the 10th of July. That is going to be How I Treat Cutaneous T-Cell Lymphoma. That's presented by Dr. Eve Gallup-Evans. So stay tuned. And that's it. Thank you very much, everybody, and have a good evening. And bye.
Thank you. Thank you.
All the very best from London. Take care. Bye-bye.
Bye-bye. Thank you very much. Bye.
Have a nice evening. Bye.
Thank you very much.